I am briefly going to discuss a practical topic, and that is the varietal screening against soil-borne pathogens by sick plot method. We already know that the plant pathogens are very diverse in terms of the part of the plant they are going to attack. Some of the pathogens like to attack the aerial parts of the plant, such as leaves, fruit, flower, or the bark, while a great majority of the pathogens which live in the soil and attack the underground parts of the plant, such as the roots or the underground stem or the bulb. The soil-borne pathogens are more dangerous because they have the ability to live for a longer period of time in the soil. And the secondly, it is very difficult to control them because it is very difficult to target them while they are living in the soil. And there is a chance that not you only kill the soil-borne pathogens by the chemicals, but also the beneficial microorganisms such as the rhizobium or the mycorrhizae. So in that context, the screening of the varieties against the soil-borne pathogen is very important. The diseases which are caused by the soil-borne pathogens include the wilt diseases or the rots, which are of various types such as the foot rot, collar rot, or the stem rot, or the damping of disease. Soil-borne pathogen usually destroy either the root system or block the xylem tissue, which result in the death of the plant or maybe the destruction of the root system. It is always remember that once the field is infected by the soil-borne pathogen, the appearance of the disease occur in the patches, unlike to the aerial pathogens, which are equally distributed among the plants growing in the field. In order to assess the resistance of the varieties against the soil-borne pathogens, we have to develop sick plot and the control plot. Sick plot is the part of the field or the land where there is a high amount of inoculum is present, while the control plot is the part of the land where there is absence of pathogen in the soil is present. So it is always remembered to develop the sick plot away from the cropping area because there is always chances that by any means the pathogen can leak from the sick plot and enter into the cropping area. And the second, it is also very important to select those part of the field as a sick plot where you have already the traces of the disease. This is very important because if the pathogen is already present, it means that the soil or the microenvironment supporting the pathogen. So these are the two conditions which usually must be met before the selection of the sick plot or the control plot. This picture will help us uh, to understand how the sick plot or the control plot is going to develop. Uh, I already mentioned that it is better to select that part of the land where you already noticed the disease in the last season. So then divide the field into two equal size plots. But it is always remember that the soil in both plot must be same. The pH, the fertility status, or the application of the irrigation, every other parameter must be same. So where you find much more disease, that is selected as the future sick plot. So the if you notice around 30 to 35% disease incidence, that part of land you can select in to develop future sick plot. And where there is comparatively less amount of disease incidence, you must select it as a control plot. So what would be the strategy? to harvest the plant on that field, and then add these infected plants into the field in order to increase the inoculum. And in the next season, you supposed to grow the known susceptible variety on that piece of land where already inoculum is present. And when you grow the susceptible variety, it promotes the disease, and the disease incidence gradually increase from 30 to 35% to 50 to 60%. And again harvest the susceptible variety and add the infected plant. So there is a build-up of inoculum. And next season, when you grow the susceptible variety, the disease incidence may up to jump to the 80 to 90%. And to the very next season, which probably the fourth year, the shows all the plant which of the susceptible variety you grow on that part of the land which we nominated as a sick plot, showing the disease symptom. It means that there is a lot of inoculum is present in the soil, and if we grow the plants in order to assess their resistance or the susceptibility, so there is a hardly any chance that these pathogens which are present in the soil may not come in contact to those plants. So that piece of the land which you develop into 3 to 4 year may be designated as a sick plot. But our strategy to develop the control plot is bit different. Here we select that part of the field where the disease incidence was comparatively low, and then we have to remove all the infected or plant after the harvesting, so that we are going to decrease the inoculum pressure. And then here we are not going to grow the susceptible variety, rather we are going to grow the non-host plant. And when we grow the non-host plant, it means that the pathogen which is present in the soil will not find the host plant and get stalled. And gradually, if we repeat that strategy again and again, the disease incidence also goes down. And finally, when we grow the most susceptible variety on that piece of land, then there we will find that no disease was shown. So that piece of land is declared as a control plot. So now we have a control plot where there is a no soil-borne pathogen is present, and we have a sick plot where there is a lot of inoculum is present. Then we grow our varieties on both plots. For example, if we want to assess the resistance or susceptibility of 4 and 5 or maybe more varieties, we grow them in replicates. And it is always remember that when we are going to grow the varieties whose resistance or susceptibility unknown, we also need to grow the most susceptible variety along them, and that susceptible variety act as a positive control in both plots. So we grow, maybe if we want to see the resistance or susceptibility of 5 varieties, we grow the 5 varieties, and then the check or the susceptible variety along them. And again, we grow 5 varieties and along with the check. And then we replicate these varieties in the plot. So, what would be the possible outcome in that scenario? The first possible outcome of this experiment is that plants of varieties and the susceptible variety, which also called check, must be healthy and disease free while growing on the control plot. The second outcome is that plants of susceptible variety must show disease symptoms while growing on the sick plot. However, response of plants of varieties may vary from the susceptibility to resistance while growing on the check plot. This simple experiment helps us to assess whether our germplasm or the varieties are resistant or the susceptible against the soil-borne pathogens.